All right, let's talk about chemistry. But for a minute, I want you to forget about beakers and lab coats. Instead, I want you to think about supercomputers. We're diving into a field that's totally revolutionizing science as we know it. It's called computational chemistry. And this isn't just a small upgrade, It's a complete digital transformation of science happening right inside the machine. So let's get into it.
For like centuries, chemistry was a very hands-on affair, right? You'd mix things, heat them up, and watch what happened. That's chemistry done in vitro or in vivo. But now there's this whole new frontier called in silico, which is really just a cool way of saying on a computer. We're trading in all those physical instruments for incredibly powerful algorithms and data. And here's the real kicker. Instead of just observing what does happen, we can now start to predict and even design what could happen.
And look, this isn't just some niche corner of science that a few people are excited about. We're talking about a field so foundational and so revolutionary that its pioneers have earned not one, but 2 Nobel Prizes in chemistry. I mean, that's a pretty clear signal that this is a massive game changer.
In fact, it's gotten so important that computational chemistry is now considered the 3rd pillar of the science. It stands shoulder to shoulder with the 2 traditional pillars. You've got experimental chemistry, that's the hands-on lab work, and theoretical chemistry, which is all the pen and paper math that describes the laws of nature. Now there's this 3rd, powerful computational pillar.
And those 2 Nobel Prizes, they really tell the story of this field's incredible rise. So, the 1998 prize? That recognized the development of quantum methods, which is basically the language computers use to speak to individual atoms. Then, fast forward to 2013, and the prize went to the folks who created multi-scale models, which let us simulate the massive, complex biological systems of life itself. It's an amazing journey from the super fundamental to the incredibly functional.
So why do we even need this 3rd pillar? Well, it's because it lets us go where traditional labs just can't. We can study chemical reactions that happen in femtoseconds. That's a millionth of a billionth of a second. We can explore the inner workings of a single enzyme or even test the properties of highly toxic materials, all without ever putting on a single pair of safety goggles.
Okay, that all sounds great in theory, but what does it actually, look like in practice? Let's get really concrete here and follow the journey of just one molecule to see how scientists use these tools to literally visualize the invisible.
Okay, so here's our challenge. This thing right here is photosystem I. It's a huge, crucial protein for photosynthesis. Its structure is stored in a public database in a file called 1kj1.pdb. But if you just open that file raw, you get this, a totally chaotic digital hairball made of thousands upon thousands of atoms. It's just pure complexity.
So the question becomes, how in the world do you even begin to make sense of this mess? Let's say our job is to study just one specific molecule hidden deep inside there, the chlorophyll molecule. How do we find that single needle in this giant digital haystack?
Well, this is where the real magic happens. Scientists load that massive data file into visualization software. Now, the cool part is the software knows the coordinates for every single atom in that mess. So with a simple command, in this case, resname CLA, they just tell the program, hey, find and show me only the atoms that belong to chlorophyll. And bam! Instantly, all that digital chaos just melts away, and you're left with a clean, beautiful, interactive 3D model of the exact molecule you were looking for. The mess becomes meaningful.
Okay, so now we can see the molecule. Pretty cool. But we can do so much more than just look at it. The next logical step is to actually interact with it, to run a digital experiment. And that brings us to a really powerful technique called molecular docking. Honestly, the best way to think about it is with a simple analogy. It's like a key fitting into a lock. The lock is our target protein, and the key is a small molecule we want to test, maybe a potential new drug. Docking software is designed to predict exactly how that key is going to fit into that lock.
The computer runs through millions, sometimes billions of possible positions and orientations to find the absolute best fit between the 2 molecules. We call it a digital handshake. But it doesn't just show us the fit, it quantifies it. It actually calculates a score that tells us how strong that interaction is likely to be.
And after all that incredibly complex calculation, the output is surprisingly simple. You get a single number. In this case, it's a Venus score of -9.2. This one little number tells us a whole story about that molecular handshake.
So, what does that number even mean? Well, it represents the binding affinity. It's basically a measure of how tightly that key sticks in the lock. And the rule is super simple.
The lower the score, the stronger the bond, and the better the fit. It's a massive, complex computation distilled down into one single powerful result.
But docking, for all its power, it gives us a static picture, right? It's like a photograph of the perfect handshake. But what if we want to see the movie? What if we want to see how these molecules actually move and wiggle and behave over time?
This gets at the really crucial difference between a computation and a simulation. See, a computation, like docking, that's designed to find a single, stable, optimized state, the best possible fit. It's a photograph. A simulation, on the other hand, models movement over time. It's a movie. It answers a totally different question. Not what is the best fit, but how does this thing actually move and function?
And this is done with an amazing technique called molecular dynamics, or MD for short. It basically applies classical physics, like Newton's laws of motion, to every single atom in the system, over and over again for billions of tiny little time steps. The result? You get a dynamic movie that shows us proteins folding into their final shape, or enzymes twisting to do their job, or drugs hitting their targets, all in simulated real time. We literally get to watch the machinery of life in action.
So we can see molecules, we can dock them together, and we can even simulate their motion. Now let's zoom back out and connect all these incredible techniques to their profound, real-world impact. I mean, how does all this stuff go from just code on a screen to actual cures?
Well, the applications are just staggering. In drug design, it's about rapidly screening millions of potential compounds on a computer to find the most promising ones, which saves an unbelievable amount of time and money. In materials science, we can design totally new materials with specific properties before anyone ever steps into a lab. And in biochemistry, it's helping us understand the molecular basis of diseases and create way more efficient industrial processes. The list just goes on and on.
So if you take one thing away from all of this, let it be this. Computational chemistry is so much more than just making pretty pictures of molecules. It really represents a fundamental shift in how we do science. It is a completely new paradigm for discovery, one that lets us ask questions and find answers in ways that were, frankly, unimaginable just a generation ago.
We're moving from a science of observing nature to a science of designing it, from discovering what is to simulating what could be. And that leaves us with a pretty fascinating final question to ponder. If we can accurately simulate the very machinery of life itself, what impossibilities are going to become possible next?